At the outset, I want to thank Dr. Vinay Gupta for inviting me to come and give a talk. A few clarification in the very beginning. First of all, you see, I'm not a clinician. I'm a basic scientist working on glaucoma genetics. And this work, currently I'm in the Academy of Scientific Innovative Research. It's a new academy. It's been created by an act of Indian Parliament. But the work, what I would describe today in general is the glaucoma genetics as we understand today in terms of juvenile disease. And that work mostly I had done when I was in Institute of Chemical Biology, Kolkata. I wasn't talking most of my work, but the disease per se as we understand. And because Dr. Gupta said that we have a little time, so I sort of try to basically explain, as we understand the genetics part of it. And I know it's a little alien to many of you, but if you are interested, please stop me at any particular point that I'm not making sense, and I'll certainly try to be able to, more clear for what I'm saying. So glaucoma, now some part of it may be a little bit repetitive, but I wouldn't do much of that, I think. Can you show me, how it works? Yeah. वही है ठीक है चलो वही करेंगे okay. And I'm color blind, so that doesn't help that very much. Maybe I should yeah, I'll just do that. So bear with me. So glaucoma is the most common cause of irreversible blindness. It affects 67 million people worldwide, the statistics says, and among them 3.1 million are blind. And as you know, there are three major subtypes of glaucoma: primary open-angle glaucoma, which is most common, primary angle-closure glaucoma, and this is the reason why I mentioned here, despite talking about juvenile open-angle glaucoma, and primary congenital glaucoma, which occurs in 1,000 live births in Western countries. It's about 3,300 live births in Andhra Pradesh in India. Other parts of India, this epidemiological study has been done so far. In Saudi Arabia, it's more common. So what it says right away that it depends on the lifestyle and pattern, the way you conduct your life. Meaning that those countries or those places, those particular community where there is more of consanguinity, there is the gene pool remains concentrated. So therefore, over the time, you have more of the same genetic disease you would find out, and that's what it is reflected here. Among these diseases, the open-angle glaucoma and congenital glaucoma, about which genetic work has just only started, not many years before, they are really called what you call is a complex disease, whereas PCG mostly is relatively simple, mostly are actually genetic disease. So in primary open-angle glaucoma, you look at it from the genetic point of view, it's a multifactorial complex disorder where both environmental and genetic factors precipitate the disease. And the incidence of POAG or primary open-angle glaucoma, we know that it increases with age. So what do we mean by this? So for example, as I I'm trying to go a little slow, that so multifactorial disease or common diseases, for example, diabetes, arthritis, any disease which is non-infectious in nature but is relatively common, you intuitively, by common sense, you can say they mostly would probably be driven by the genetic defects as well as environmental factor. You pick any disease. The extent of genetic contribution might vary, but it would be normally combination of both. And it would not be exclusively for one factor. Sometimes yes, I'll show you example, but mostly not. That makes it so difficult because we have understood the molecular basis as to how gene works, the Mendelian's pattern, we understand that, how it works. But whenever you combine even two genetic factors together influencing a pathological situation, the situation already becomes difficult. You can understand that if there are many more factors, lot more difficult it would be. So that really was the challenging situation. Now we will see that in the second point, it said that primary open-angle glaucoma cases have some familial component. That is 72%. but it rarely follows Mendelian pattern of inheritance. So what does that mean? So that means that whenever you as a clinician would go and ask the relevant question that does it run in your family, what is reported that a vast majority of the patient would say that in their family they have the disease. So how would you interpret it? Unless if the family members lives in different places and if they are not entirely influenced by environmental factors, then the genetic factor is what really remains. So therefore you would simply by the fact, even without doing any genetic analysis, you can say they have a genetic predisposition for the disease. So in case of primary open-angle glaucoma, so far whatever study has been done suggests that a large portion of those are actually probably has some effect of genetics. But the challenge is, if you know a little bit of genetics, that it does not follow Mendelian pattern of inheritance. What, for example, primary congenital disease, congenital glaucoma does. So therefore, if you are treating with, for example, primary congenital glaucoma, or for that matter, if for a minute you go away from glaucoma and talk about retinitis pigmentosa, which is 100% is genetics. So there are 40 different loci, but whoever have the disease, if the gene is identified, you can predict with very high confidence that who is going to come down with the disease. So that can happen in case also primary congenital glaucoma, you don't have that opportunity because that's congenital. But that's really what is meant by having a specific pattern that follows the Mendelian pattern of inheritance, we say. However, the today's topic, juvenile open-angle glaucoma, is frequently passed down through families as an autosomal trait. So in those cases in juvenile open-angle glaucoma where the familial component has been found out, even without doing any genetic analysis, just by looking into the pedigree, if you happen to have a pedigree in which you have multi-generational, better 2 than 3 generation, it's better if you have multiple like siblings, then even without doing any genetics, just by following the basic principle of genetics, you would be able to tell whether it is autosomal dominant or it is recessive. Now if I'm not making sense, to make a little more sense for some of you, autosomal dominant means that in the genes we have 2 copies, 2 alleles. When it is defective in only one allele and still cause the disease, that is called dominant. Whereas when there is both copies of the defective gene are required to precipitate the disease, then it is called recessive. So therefore, if somebody gets a defective gene, one would get the disease for sure in case of autosomal dominant disease. But if you think about societally, recessive diseases are a lot more challenging because they slip in the society. If you think about, for example, you go to the Western world, the cystic fibrosis, about which people didn't know for a much longer time, and it's a huge gene. The discovery was found out later that it was really a founder mutation that through generations after generation after generation sat, and then it ballooned, and people started finding the disease in many places. Why? Because it, as I said, that you may be a carrier for a defective gene, but you are not having the disease. If the disease is of recessive nature. In case of juvenile open-angle glaucoma, in whatever cases have been found, it mostly have been found out to be dominant. So for its recognition, its finding out in a family is relatively lot more easy. Now, if you look into the primacy, as my previous speakers have said, that juvenile open-angle glaucoma is open-angle glaucoma, it just occurs in relatively early stage. So far, a lot of work has been done on primary open-angle glaucoma from the genetics point of view, and it has also been found out, and they have given us, so whenever genetic work is done, people don't know where it is, they just know somewhere in a specific region that you identify. So it's given a chromosomal location. So if, for example, in the very first example, you say 1q21 to 31, means it is chromosome 1. It's in the smaller, shorter arm of the chromosome, the longer arm of the chromosome, and it gives its coordinates. Like whenever you drive, you have certain coordinates. It just gives you coordinates. So there are millions of bases. But you see that there is only few genes. In the last column, the gene names are given where the gene has been identified. So that really shows the myth in the fact that when in 2001 the Human Genome Project was completed, there was a big news about this Francis Collin and Craig Venter coming up with the Human Genome Project. We thought we are going to solve all the problem. Many years have passed. Still, you will find out in any meeting you go where the genetic work is going on, you still find that these cases, lot of the space are empty, meaning that in those cases the gene has not been identified. Why is that? This is not because they are multifactorial. If these loci have been mean that for in every single case, in every single those cases, you open the paper, you see that has been based on specific family in which the disease have been found out, the gene has been hunted and has been located to a specific location. The reason for it is that we don't still, and I'm sure you will not be maybe so much interested, so I won't go more in that we still do not understand much of the, even though we have the entire human genome, but we do not understand a lot about the genes, still discoveries are being made as to which part of the genome we knew those to be as the junk DNA. Lot of those are currently sort of making sense. So, of course, what your mind does not know, eyes don't see. So, by the knowledge of the what you know is in your mind. So if you do not know that what really are the functionality of some of the differences that you found out, you will not make the discovery. So we are not there yet. That is the reason you see there are so many empty spaces. So you see that in case of glaucoma, among so many different loci that is listed, only myocilin, optineurin, ASB10, WDR36 and NTF4 are the ones which has been associated with the disease. Now the point I am trying to make here, I think this would be relevant for you, even for your practice, is that when we talk about myocilin, like my previous speaker said, about associated with juvenile open-angle glaucoma, it does not mean that it always would be only in juvenile open-angle glaucoma. You may have a defect in myocilin gene, but depending on where the defect lies, it will be either manifest as juvenile disease or as an adult disease. It can very well be that there is a defect in the myocilin gene itself, but the defect is not causing the juvenile form of the disease. Okay, so there is a lot layer of complexity is there, and I can tell you what other loci you have seen. There have been lot of expectation, hopes when these other loci has been discovered. But if you go and look into the human genetic disease database, you will find out that the contribution of the myocilin gene is much higher relative to the other loci that has been discovered, which basically says there is a lot more heterogeneity in the disease, meaning with the big centers. What's kind of disappointing, you see, that despite so many loci, so much genetic work, so many publications being made, you see that genetic bases have been identified only in 5% of the cases. In many cases, we do not know the genetic basis yet, which means that that doesn't mean that the genes are not involved. It only means that a lot more work still needs to be done. I'm not so sure that in this audience I can go too much into the depth into the genetics, so I don't intend to unless you ask me a specific question. But I would just say that the next step towards the genetic from going from basic genetics for doing linkage analysis in case of specific families, what have been contemplated and have been attempted to do, particularly in case of common diseases or complex diseases, is what is called genome-wide association study. Because now we know the entire human genome sequence, therefore instead of making a fishing expedition here and there, the idea has been to go and look for the entire genome for any particular defect. So those studies has also been done in case of primary open-angle glaucoma, but I am not aware of any study being done specifically for juvenile open-angle glaucoma. This is a slide which me and my student had actually done quite a few years before. We published this as a review. I asked my students simply to find out how many papers you find in which different factors that have been shown to be or associated with, have been claimed to be associated with the primary open angle glaucoma based on genetic studies, and they have been explained by participating in specific pathway. Now clearly I don't expect to read anything about in this entire slide at all. The only point I am trying to make, that unlike retinitis pigmentosa, in which case caused by multiple genetic factors, the diseases are primarily known to be caused by defect in the phototransduction pathway. So you know that the light, for processing of light, it goes from pathway from one end to the other, and in that passage, if anywhere is the defect, that might snap the link. That's pretty intuitive. You simply go and try to discover it, those things, right? So I sort of, it's a little bit oversimplified, but conceptually that's what it is. But the reason I'm trying to put this slide here just to show you the level of complexity that is there in primary open angle glaucoma, and we are all basically nibbling in one particular point here and there. So over the time we expect that we would understand the network better and we'll understand the disease better. One thing I like to say, even if my time finished and if I had to finish my talk, Dr. Binay said, okay, right. So that was my indirect, so 10 minutes more. So we'll finish in 10 minutes. So one thing I would say, if you would have said that, Kunal, could you finish it in 1 minute, then I wouldn't go with my slides at all. The only message that I'll try to give to all the clinicians is that the entire work on genetics is extremely, extremely important, that it is dependent on collection of the samples with very detailed clinical phenotype. It is extremely important. And even if you ever not, see for example in your center, if you are not working at all with genetics, you don't even thinking about working in genetics, but if you really carefully collect the tissue sample and keep it for a long period of time, that could really turn out to be a gold mine for somebody else comes later. That exactly what has happened in Harvard many years before, when Ted Drija came and joined there, and Elliott has kept, he was a fantastic clinician, but he had no idea about any genetics at all. But he has this posterity and thinking, and he knew that because it is a genetic disease, someday I will get someone and someone will do it. So he kept these samples, the blood sample at that point, in the freezer, and when Ted Drija came as a young MD with the knowledge of genetics, started working one after other, all the genetic loci started coming, So what I'm basically trying to tell, that whether you are interested in genetics or not, but if you are willing to treat your patients and get the genetic information is helpful, you may get help from others, and the way you can help is simply by collecting those samples. I myself have spent 12 years collecting samples from different diseases I work, and when I did not working on a high gear, my students are using the samples, but I wasted that amount of time just for getting it. So this point I think is very important that I wanted to basically tell. So now going on to the specific topic, so juvenile open angle glaucoma, I don't think I have to give any introduction. This has been well given already. Now for genetics, what is known? The studies suggest that it's an autosomal dominant pattern of inheritance, but I would tell that mostly that one does not know that entirely that's really true. Myocilin is estimated to be responsible for about 33% of the juvenile open angle glaucoma. In addition to that, CYP1B1 gene, which by itself exclusively causes primary congenital glaucoma, in combination with myocilin can expedite the disease. It has been shown very, very clearly in a publication in American Society of Human Genetics, and later in different parts of the world, including our lab, we have seen that whenever you have a defect only in, say, myocilin gene, it has been found out, and the particular family I'm talking about, when the work which was published in American Society of Human Genetics, that the myocilin defect was present in a family which causes the disease around 50 years of age. But whenever there is a defect in CYP1B1 gene, which by itself causes primary congenital glaucoma, combination of having two mutations together, the disease, the patient's, the disease started at the juvenile stage. It was around 25 years or so. So, so therefore, you have to remember that also, that this can also happen. Involvement of other genes in POAG appears to be more restricted based on the investigation that has been done so far. So here are the name of the loci, and there's no point of going through the letters. It basically shows that few other loci have also been found to be causing juvenile open-angle glaucoma. And if you see anybody tells you loci, that means all the work must have been done based on all familial samples. This is just an example that we had worked on it many years before. This is a family of primary open-angle glaucoma with a mutation that is written as Pro370. Means that at the 370th amino acid, the proline has been replaced with the leucine that we found out. And this specific mutation, and this was done in Calcutta. And we found that in many patients, this family has many patients, as you see, and because it's in a village, because socio-economically they are very weak, then by very early of age, they were almost blind. So we worked on this family and we basically found out, and the symbol, as you see, the black symbols means that those are who are affected, and the blue symbols are those whose age are relatively less, but they are simply by genetic analysis, we found the specific genetic defect and we could predict that those individuals would come down with disease. And in this specific case, as I'll tell you in a few minutes, that this specific mutation has been studied across the globe in many different countries, and it's very highly penetrant irrespective of any ethnic group. So if your patient have this mutation, be rest assured that that person probably is going to come down with the disease. If you and it runs in a family and it works pretty well from that perspective. So for anywhere wherever you work for genetics, usually obviously the point is, what's the point of work of genetics unless you can take advantage of it and you can relate to phenotype. So obviously whenever we work on it, it really depends on the heterogeneity. And I think the major reason why in case of glaucoma we have not been able to make major strides is because in glaucoma there lies a very tremendous amount of heterogeneity. Until we are able to dissect the heterogeneity, it would be very difficult for us to identify the genetic loci and understand it. So here it is, I'll just read it out, that the known genetic variations can act as an important tool for screening relatives to JOAG patients. Genetic analysis of variations can be used as predictor for such specific phenotypic characteristics related to the disease. Such information can be handy, especially for JOAG cases. So there are two important scenarios you can think where we can use this genetic information. One is to predict the disease progression, and second, to predict the response of specific drug. So first is the predictor of the disease progression. You say a recent report for juvenile open-angle glaucoma advocates the importance of genetic testing, especially in children. Well, we already know that. If you follow the table below, you see that the table represents an example of association of myocilin variations and disease phenotype, which I was saying a few minutes ago. In this case, on the left-hand side, left column of the table, you see that there are just simply different mutations. We don't have to go by the name. Just different mutation, and see that depending on the diagnosis, age and the highest IOP, the risk factor varies, whether it will be mild risk or it will be intermediate risk or it will be severe risk. So if you have an unequivocally a genetic loci you have identified in families, enough work is done, then I think you can actually do this kind of risk assessment very well. Currently percentage certainly is very low, as we know for many other diseases as well. So with respect to the prediction of the drug response, the inter-individual genetic variability is known to cause of adverse drug response in various diseases, but in case of glaucoma, only few such studies have been performed. So the following are the examples which suggest the importance of carrying out pharmacogenetic studies and their integration into clinical practice. So what you see here is basically SNP means the single nucleotide polymorphism. Variations in the gene, they really are not mutations. Those are otherwise innocuous, present in our gene, and depending on the genotype, meaning the two alleles that we have, based on their character and the gene, the different responses depend. So here, for example, you see the first response in case of ADRB2 gene in a specific SNP is the drug is like a beta blocker, and that it works in a positive response in the background of certain genotype. In case of CYP2D6 gene, you have bradycardia or you do not have it, depending on the genotype when you use the timolol drug. So this is not, as I said, not much work has been done with respect to juvenile open angle. Lot of work has been done, but I would say has not more information, much of is known. And I think I can stop. Bina, you could stop me at any point now. The rest of the few minutes, if I would like to say, is that the huge advancement that has done with respect to after the Human Genome Project has been completed and the challenges of deciphering and working on it, new ideas has come, and exome sequencing is one such idea was being used to expedite our hunt for defective gene. I don't think we would have time to discuss this more and maybe not be appropriate forum, but this is one of the things that we do. Genetic studies have been done following this strategy in case of juvenile open angle glaucoma and primary open angle glaucoma, but now some loci have been identified, but I don't think you'd be interested more than that what I said. The one other point, I think, because I think this is for the learning session, I think these two quick slides would be useful, that recent thoughts on missing heritability. So most genetic variants identified confer only small increment in the risk and explain a small portion of familial clustering. De novo mutations are being found to play important role in rare as well as common diseases. These de novo mutations may significantly contribute heritable component of complex diseases, which are transparent to finding by GWAS. Now the question is, this is the point, that while the question of missing heritability is being addressed for other diseases, it is not yet a major focus of ocular disease. In case of ocular research, who are doing ocular research, they should really, I think, should catch up on this very important area that has come, which basically this slide says, and I will not explain in detail, is that whenever we talk about genetics, we always think in terms of germline, and we will always think that the defect that occurs in the germline. But now the thought is that actually the somatic cell mutations, which was always being attributed only to cancer genetics, has been now attributed to many of the other diseases, major diseases which are known as genetics. And here are just some examples, like for BAC1 gene variation in abdominal aortic aneurysm, and other examples are there. So which shows that now looking into the somatic mutation is also very important. Why I brought up this point? Because this is something who are doing in ocular research, interest in genetics. This is an area that is unexplored to be what is very important thought that has come with respect to finding out the missing heritability, which really occupies the major area of our study, that part is missing. So for finding the missing heritability, detection of total genomic load of detrimental variations can be found in patients where, for example, those are inherited. In case of non-inherited diseases, detection of somatic mutations unique to the disease tissue obtained from the patient for both bilateral and unilateral expression of phenotype would be important, and the de novo mutations could be segregated as germline or somatic mutation if the trios are available. I think I should stop there. I don't, I don't know whether I did more than what was needed, but yeah.